Right, this is Dr. Philip Zak, clinical geneticist, and this is the third of 3 lectures to help you pass the genetics questions for the MRCP CH1 exam. In this lecture I'm going to talk about the mechanisms of inheritance, because this is quite a common either theme on its own for questions or part of other questions. So in this lecture we're going to talk about having a basic understanding of the mechanisms of inheritance, which of course are chromosomal, single gene, which can be dominant, recessive on the autosomal chromosomes, or recessive or dominant on the X chromosome, mitochondrial, and it's important to be able to recognise the different patterns of inheritance, and of course know which human disorders are inherited in which manner. So it's important to have a basic understanding of the structure and function of the genetic mechanisms in the cell. And this simple illustration shows you the relationship between the chromosomes in the nucleus, which can be seen down the microscope, and the DNA packaged within the chromosomes and its base pair sequence. And it's important that you have a basic understanding of how genetic information flows not only from generation to generation, but is also used within the cell to enable the cell to function. Now then, people often ask, how can I learn which conditions, which diseases, are inherited in which way? Now there isn't an easy answer to this. There's no simple formula to allow you to work out which condition is inherited in which way. However, my approach would be one of exclusion. As you're learning genetic conditions, learn those, first of all, which are on the short lists. The short lists are conditions inherited in a mitochondrial fashion, and that's a short list because the mitochondrial genome is a very short genome. There are only a few genes that can go wrong. Then learn the X-linked dominant disorders. This is a short list because very few genes on the X chromosome are inherited in a dominant fashion. There is a rather longer list of X-linked disorders that are inherited to recessives, but it's a relatively limited list because, of course, it's only one chromosome. I would then suggest that you learn some of the commoner chromosomal disorders. Make sure that you know that they're chromosomal rather than single gene disorders. And then, if a condition comes up that you haven't heard of, then it's unlikely to be on any of these short lists. And so the chances are it's an autosomal condition which will be either dominant or recessive. So let's start off with mitochondrial disorders. Simply because it's a short list, and we can therefore get it out of the way quite quickly. Now, as I said, the mitochondrial genome is a short genome. It's a ring of DNA about 16,000 base pairs around. There's only a small number of genes on it, and most of those genes code for transfer RNAs rather than proteins. So there are only 13 genes on the mitochondrial genome which actually encode proteins, and so there's a limited amount of damage that can occur. And here is a pretty complete list. I can't say it's absolutely perfect, but it's a pretty complete list of pretty much all of the conditions you're likely to come across that have mitochondrial inheritance. So if you see a condition on this list, the chances are it's mitochondrial inheritance, except for the last one, Lee syndrome, which can also be inherited as a recessive trait due to mutations, of course, in the ordinary chromosomes. Genes on the X chromosome are usually inherited as recessive traits. However, a small number of them are inherited as X-linked dominant traits. Now again, I'm not claiming this is an absolutely exclusive list, but it's probably most of the common conditions that you're likely to come across. So here are most of the X-linked dominant conditions that you come across in your practice or in the exam. One I'd particularly draw your attention to is Alport syndrome, which again can also be inherited as a recessive trait. But apart from that, most of the others are X-linked dominants. Right, so let's move on to start the questions. First question is a straightforward one, asking you which of the following show recessive pattern of inheritance, autosomal recessive pattern of inheritance. So look down the list and simply make your choice now. I suggest you write down the answers on a piece of paper so that you can learn from your mistakes. Okay, all of these except C are inherited as autosomal recessive conditions. Of course, C, Edwards syndrome, is due to trisomy 18, a chromosomal disorder. Now, autosomal recessive inheritance occurs when one needs 2 copies of a mutated gene in the same individual in order for the clinical features of the condition to manifest. And so in order for two copies of the mutated gene to end up in one individual, that individual has to inherit it from both their parents. This means that one typically sees in a family tree what's called a horizontal pattern of inheritance. It tends to affect people in the same generation. The common pattern is that we see siblings affected by the same condition, but in this case we see cousins affected in a consanguineous family. Now the double lines indicate consanguinity, i.e. marriage between people who are related. Now of course the reason why this matters in a recessive condition, particularly a rare recessive condition, is that because in order for a rare mutation to meet up with a copy of a rare mutation in the same gene, this is a very unlikely event to happen just by chance in the general population. But it's much more likely to happen if two individuals from the same family who share genetic background have offspring. In that situation, the one rare mutation can come down the family tree in two directions around from one side and the other side, and then meet itself again in the offspring. And this is what's happened in this situation. A mutation will have come from the great-grandparents or beyond, down this family tree, two ways down the family tree, and then when cousins marry, the rare mutation is able to meet back up with itself in one individual, leading to a recessively inherited trait manifesting itself. Let's try question number 2 then. This asks about autosomal dominant inheritance. Which of these conditions shows autosomal dominant inheritance? Write down your answer now. In this list, all except A, congenital adrenal hyperplasia, show autosomal dominant inheritance. You'll note that adult polycystic kidney disease is a dominant condition, whereas recessive infantile polycystic kidney disease is a recessive condition. So here is the list again, and of course congenital adrenal hyperplasia is an autosomal recessive condition. Dominant inheritance tends to show the opposite pattern to recessive. It tends to show a vertical pattern of inheritance, going down from generation to generation. And that's because dominant inheritance means that one only needs a single copy of the mutated gene in order to show features clinically. So in this situation, in the top generation, we have a brother and sister. The sister then has a number of children and passes it on to approximately half of the children. They then pass it on down to their children. But there's something amiss with this family tree. Can you spot what it is? Hopefully, you've noticed that in the second generation, the second individual from the left, a circle indicating a female, appears to be unaffected by the condition, and yet she has gone on to have two affected children, a boy and a girl. So how can this have happened in a dominant family tree? Well, in fact, this does happen relatively commonly in dominant family trees. Now, there are a number of explanations for this, but the most likely is reduced penetrance. And this is thought to be due to the influence of other factors apart from the mutated gene, such as the other genes, or, in certain conditions, environmental factors, which means that an individual may inherit the mutated gene but not show clinical signs. Penetrance is an all or nothing event. A person either shows clinical signs or they do not. It's different from expressivity or expression. Expressivity, variable expression of a gene, is where the degree of severity of the manifestations vary from person to person, whereas penetrance is a situation where a person shows absolutely no signs of the condition whatsoever. It's relatively common in dominant conditions. It's the most likely explanation of this family tree. Right, let's move on to part 3, question 3. Now this is asking about a rather unusual form of inheritance pattern. Not an inheritance pattern as such, but it's a molecular mechanism. Trinucleotide repeats. Which of these conditions is associated with this molecular mechanism? Mark your answers now. Okay. In fact, all of these are associated with trinucleotide repeat sequences. A trinucleotide repeat is precisely what it says. It's a sequence of DNA in which a trinucleotide, a section of 3 nucleotides, is repeated over and over and over again. In this instance, it's the CAG repeat, which is seen in Huntington's disease. There are a very limited number of conditions that are due to expansion of trinucleotide repeats. The difficulty occurs because trinucleotide repeats, once they get to a certain length, become genetically unstable and are more likely to expand to a greater length with each generation. The degree of this uncertainty varies from parent to parent. In some conditions it's more common in the father and others in the mother. Once the trinucleotide expansion gets beyond a certain point, it interferes with the normal function of the gene and therefore produces clinical symptoms. The actual length of the trinucleotide that's required to interfere with the gene function varies from gene to gene. The majority of trinucleotide repeat conditions are inherited as autosomal dominants, but there are a couple of exceptions for you to remember. One common one is Fragile X, which of course is X-linked. The other is Friedreich ataxia, which is an autosomal recessive condition. Question 4. A gene defect in a specific chromosome can be identified for which of the following? Now, with this question, think carefully about the wording of the question and what it is actually asking you. Then write down the answers you think are correct. In this question, A, C and D are correct. So you may be a little confused about why E is not included. Let me explain. The wording of this question asks for specific gene defects in a chromosome, not a particular chromosome defect. I.e., the question is actually asking which of these are single gene disorders. So when you have a question that you're not entirely sure of its intention, usually the intention is the simplest face value meaning of the words. Don't try and read more into a question than is intended. Now, in this particular question, sickle cell disease is of course due to mutations in the beta-globin gene, usually the HBS mutation. Cystic fibrosis is due to mutations in the CFTR gene on chromosome 7. Neurofibromatosis may have caused you a little confusion because of course there are two diseases called neurofibromatosis, or NF. The much commoner, NF1, is due to mutations in chromosome 17 in the neurofibromin gene, and the much rarer type 2 is a completely different condition due to mutations in a different gene, the Merlin gene, in chromosome 22q. However, for the purposes of the question, it doesn't matter which NF they're talking about, because they are both single gene disorders. A myelomeningocele, of course, does not have a specific gene locus and is probably a multifactorial etiology. And the one which may have thrown you is Klinefelter syndrome, which is a chromosomal disorder, which is of course due to the influence of multiple genes on the X chromosome, because of course all the chromosomes along the entire X chromosome are duplicated when you have an XXY phenotype. However, you may of course remember that normally when there's more than one X chromosome in a cell, one of the two X chromosomes is inactivated. And so it may have occurred to you to wonder, if a normal woman with 46 chromosomes and 2 X's normally inactivates one X chromosome, why does a woman with only one X chromosome, with Turner syndrome, have any symptoms whatsoever? Because surely she's in exactly the same position genetically as a normal woman. And similarly, you might ask, why would a Klinefelter man, who again would inactivate one of the two X chromosomes, be any different from a man with 46XY? The answer to this is that inactivation does not actually affect the whole of the X chromosome. There are a few small regions in the X chromosome called the pseudo-autosomal regions, or PAR regions, which are not inactivated. They escape inactivation because these are regions which are homologous to regions on the Y chromosome. They're very small regions, they only contain a few genes, but recombination does occur between the X and the Y chromosome at these regions. And because these regions are not inactivated, a woman with Turner syndrome will have smaller number of genes in the pseudosomal region active than a normal woman. And in particular, the SHOX gene, SHOX, is thought to be responsible for the short stature in Turner syndrome. Similarly, a man with XXY will have an additional X chromosome and will therefore have duplicated genes in the pseudoautosomal region, which is presumably responsible for most of the effects of Klinefelter syndrome. Question 5. This question asks you to identify disorders due to having an extra chromosome. There's a word for this, these are aneuploidies. Which of these conditions are aneuploidies? Mark them down now on your piece of paper. And here are the correct answers. C, Turner syndrome, is of course an aneuploidy, but it's not due to an extra chromosome, it's due to a chromosome missing. The other three, A, D and E, are due to extra chromosomes 21, X and 13, as shown. Angelman syndrome has more than one etiology, but it's basically in an imprinted region where one may have a deletion or uniparental disomy. Now, trisomy 21, Down syndrome, is of course always caused by an extra copy of chromosome 21. But it's important to remember there are three ways in which this event can happen. In the majority of cases, 19 times out of 20, this occurs due to non-disjunction of the chromosomes. That is, the chromosomes fail to separate normally at meiosis, when the egg or sperm has been produced, and so two chromosomes, 21, are found in the egg or sperm, which is then fertilised, and a cell with 3 chromosome 21 results. In this situation, there is an increased recurrence risk, but the increase is only to a level around 1%. So couples that have a child with non-disjunction Down syndrome should be advised that there is a small increased risk of recurrence in future children, but that this risk is only around the 1% level. However, there are a couple of other ways that Down syndrome can result, and these are due to structural rearrangements of the chromosomes. In around 3 or 4% of cases of Down syndrome, there is a Robertsonian translocation between chromosome 21 and another acrocentric chromosome, usually 13 or 14. In this situation, the chromosome 21 is physically joined on to a chromosome 13 or 14 at the centromere, which means that these chromosomes cannot separate at meiosis. They have to go together into the one cell or the other. What this means is that there is an increased recurrence risk above that of a non-disjunction event. With a Robertsonian translocation, depending on whether it's carried by the male or female, the recurrence risk is either 10 to 15% for a maternally derived Robertsonian, or a 2 to 5% recurrence risk for a paternally derived Robertsonian translocation. In a small number of cases, around 1% of Down syndrome cases, there is, however, another form of translocation. This is a Robertsonian translocation which produces an isochromosome, a chromosome 21 joined to another chromosome 21. Now, when these chromosomes attempt to undergo meiosis, there is no way that the two chromosome 21s can separate because they're physically joined, which means that they will either go both into one cell, both into the other cell, which means that the daughter cells will either have 2 chromosome 21s or none at all. The resulting cells following fertilisation are a monosomy 21, which usually results in a miscarriage, or Down syndrome, which means that all the live-born offspring of an individual with a 21-21 translocation must have Down syndrome. So it's important to recognise these rare but high-risk scenarios when counselling people with Down syndrome children. Now let's move on. Question 6 is some more questions about chromosomes. But I'll warn you in advance on this one. This is a negatively worded question. So you need to understand which of these stems is correct, but then reverse your answer, because the question is asking which of these is incorrect. So go through and decide which of these statements is incorrect. Write down your answers. In this situation, C and E are incorrect. The pairs of autosomal chromosomes are normally arranged from largest to smallest in a karyotype. Question E is incorrect because although the commonest sample for fetal chromosome analysis used to be amniotic fluid, it is now chorionic villus sample, which is more commonly used because samples can be taken earlier in pregnancy. So as I said, beware, this is a negative question, and always look out for negative questions when they arise because they can throw you unnecessarily. Here is a karyotype on the right side of this slide. The chromosomes have been arranged from largest, chromosome 1, down to the smallest, chromosome 22, and then the sex chromosomes X and Y are placed at the bottom right of the picture. The simplest way this is done is simply by taking a photograph of the chromosomes down a microscope and taking a pair of scissors and cutting them out, although nowadays this is normally done electronically. Looking at an individual chromosome, the picture on the left, we see that chromosomes have a characteristic structure. All chromosomes have a centromere. They have to have a centromere in order for them to participate in mitosis or meiosis. Chromosomes that don't have a centromere are lost with each cell division because they can't separate at meiosis or mitosis. Most chromosomes have two arms, a short arm, P, and the long arm, the Q, although a few of them only have one arm, which is usually designated the Q or long arm. These are acrocentric chromosomes, which are 13, 14, 15, 21 and 22. These are the chromosomes which can participate in Robertsonian translocations. The other chromosomes can participate in reciprocal translocations, where they swap portions of an arm. Incidentally, the reason for the designation of short and long arms as P and Q is historical. Some of the earliest research work done on chromosome analysis, chromosome structure, was done in France. And there the chromosome arms were named by the French names the petit bras and the grand bras. For some reason over the years in the literature, the P for petit was retained, but the G for grand was turned round into a Q, and so now we have the P arm and the Q arm. Chromosomes can be analysed from practically any mitotically active cellular tissue, but the commonest ones which are used in practice are blood, amniotic fluid, chorionic villus samples, and also bone marrows and sometimes skin fibroblasts. Question 7. Which of the following are correct about X-linked recessive inheritance? Look through the options and make your choice. Write down now what you think are the correct answers. In this situation, A, B and D are the correct answers. C is incorrect because we know that in a minority of cases, women who carry X-linked recessive diseases are what are known as manifesting carriers. They do show mild signs of the condition. And of course, E is incorrect simply because CF is a autosomal recessive condition. This is the typical pattern we see in X-linked inheritance, and it's given the name Knight's move inheritance after the chess piece, the knight, which moves in an L-shaped move on the board. A chess piece, the knight, moves by jumping over a square without touching it and then landing on a square at the end of its L-shaped move. And this is the typical pattern we see in a family tree, where we have affected males separated by unaffected females. In this family tree you can see that in the top generation one has an affected man who has a sister who appears unaffected, but then she has an affected son. She also has an unaffected daughter, who then in the bottom generation herself has an affected son. These two women therefore, the mother in the top generation and the woman on the left-hand side of the second generation, are both going to be obligate carriers for this X-linked condition. They must be carriers because they have passed it on to their sons. Question 8. This question asks you to match the carrier types to the syndromes. And this is a question of how well you know your chromosomal disorders. So look down the list and write down for each condition which is the correct carrier type. First one, Down syndrome, is of course due to trisomy 21, and so 47XX plus 21 will be a correct karyotype. type. Klinefelter syndrome is due to an extra X chromosome in a male, and so is XXY. The diametric opposite this is Turner syndrome, in which there is a missing X chromosome in a female. This is either written as 45XO or simply 45X. The O simply indicates an absence of a chromosome. Patau syndrome is a severe condition which is resulting from an extra chromosome number 13, and of course can occur in either males or females. Edwards syndrome, similar severe condition, is due to an extra chromosome 13, trisomy 18. The condition XXX is, as the name suggests, due to 3 copies of chromosome X. And XYY is a condition which is relatively unknown but quite common, And the difficulty in the exam is that people sometimes confuse Klinefelter and XYY simply by reversing the number of X's and Y's. So just be careful not to get those two confused. Question 9. This question is about X-linked recessive disorders. So have a look through the options and see which you think are the correct answers. Mark them down on your paper now. Okay, B, C and D are the correct answers. And finally, let's take a slightly more clinical example and see how this can be used in a more clinical scenario. Here's a couple who've come to see you about their second son, John, who had Down syndrome. Their first son's unaffected. Now they're concerned because there's a strong family history of Down syndrome. One of John's cousins is also affected, as well as Mrs. Smith's brother. Mrs. Smith's concerned about drinking a lot of alcohol during pregnancy. So which one of these do you think is the most likely cause of Down syndrome in the family? Choose one now and write it down. The most likely answer in this scenario is B. So let's think about why that is. As you know, Down syndrome results from an extra chromosome 21, which is usually the result of a non-disjunction event at meiosis, failure of chromosome separation. However, when you have a situation where there's a recurrence in a family, then it makes you think that there's something other than non-disjunction occurring. Of course, the family could just be unlucky and have two individuals by two non-disjunction events. But it's more likely that there is a structural chromosome abnormality being inherited in the family, and this is likely to be a translocation. Now, a Robertsonian translocation is the name given to the fusion of two acrocentric chromosomes, that are chromosomes that only have one arm. When these chromosomes fuse, they form what looks like a metacentric chromosome with two arms, and the acrocentrics are 13, 14, 15, 21, and 22 in humans. In the case of chromosome 21, producing a Down syndrome risk, the commonest Robertsonian translocation is a 14-21 translocation. Now, you might have tried answering C, an isochromosome, with two chromosome 21s stuck together. In this situation, the two chromosome 21s cannot separate at meiosis. Every single offspring has to have Down syndrome. But you were told in this scenario that the couple had had a previously unaffected child, which means that an isochromosome 21 is unlikely to be the answer here. Prenatal exposure to alcohol, of course, is not a good idea for any woman, but it's not a cause of Down syndrome, and so that's not the answer. The most likely explanation in this case is a Robertsonian translocation, probably between 14 and 21 or possibly between 13 and 21, which has been inherited through the family and is resulting in a high risk of children having Down syndrome. So, I've taken you through some of the common mechanisms of inheritance in human conditions, talked about a number of chromosomal disorders, taken you through the patterns of inheritance of single gene disorders. We haven't mentioned mitochondrial because this is quite an uncommon form of inheritance, but I would advise you just to read up on it in your own time. It's important to recognise the patterns of inheritance as they appear in family trees, or in the exam sometimes the descriptions of family trees, and to be able to recognise the typical patterns of horizontal inheritance in recessive family trees, vertical inheritance, possibly with reduced penetrance in dominant family trees, and the typical Knight's move pattern in X-linked recessive family trees. And it's important to try and have some basic knowledge of how individual human disorders are inherited. Thank you.